So this was the last image we have seen in our previous class, in which we saw that you could associate these chemical modification to some function which they are performing. Uh, so we have seen this picture in which we have seen that you could want, if you are interested to see the role of acetylation in cell cycle, so you could arrange an experiment like this where you could try to make uh, different settings where you can stain the DNA using this stain which is called Dappy stain, and you could also stain acetylation using this acetylated antibody against histone H3, and you could see that both of these stains are giving almost the same arrangement of color pattern, wherever there is a DNA present, there is an acetylation going on there, and this correlates very well with different cell cycle stages, and this tells you a thing that acetylation is doing a role doing a very important role in different cell cycle progressions. So if you are going to stop this acetylation, that means you are going to somehow stop progressing of the cell, so the normal working of the cell will be severely damaged. So it might be possible that cell is no more dividing. So this is what, so on this panel we haven't discussed that last time, but let's quickly have an impression about this panel. So this tells you the role of acetylation in spermatogenesis, and it has been seen with lots of reported studies which tells that histone acetylation plays a very important role in spermatogenesis process. So you could see this arrow which tells of soon after meiosis there are a number of acetylations which are happening on histone, and not only the acetylation. For example, this is the lysine 9 methylation, and also as well as the serine 10 methylation, serine 10 phosphorylation. They are also playing a very vital role in spermatogenesis process. This overall tells you that the role of chemical modification cannot be ruled out in normal uh, developmental processes. So this tells you, and this is very important, that we need to understand that histone modification is in fact a separate entity which somehow take the advantage of being, I mean, sometimes we could consider them as a code. We are going to discuss them one by one. Just let's first discuss what techniques you could use to study histone modifications. This lecture is very important for you people because this will give you an overall scenario of different chemical strategies which you could opt in order to elucidate different chemical modifications on histone. So, some of them are very famous one. For example, you could have heard about the chip techniques. These are called chromatin immunoprecipitation techniques. So uh, uh, do not mix it with another chip which is written by all small letter, which is called chip. We are going to come there. So this technique, which is called chromatin immunoprecipitation technique, is a strategy which could be used to elucidate localized histone marks. So that means the histone marks, you know them. For example, this is for the known histone marks. For example, you are interested in an acetylation at lysine 4 of histone 3. So you know that there is a lysine acetylation going on at histone 3 at lysine 4. So you have localized this position. So you could go for this technique. I mean, it's not that specific. You could also go for native histone protein. For example, you could make a chip technique using uh, just the histone H3 antibody. So this is the still it will be a chip technique because all those techniques which are focusing on chromatin precipitation, you could say them, these are chip technique, these are chromatin immunoprecipitation technique. In fact, this is a sub branch of a bigger technique or umbrella technique which is called immunoprecipitation technique, which employ the strategy of antigen antibody reaction. So in our normal cells we have something like this, where you have an antigen and there is an antibody which could specifically bind to this antigen, and by by their reaction, by their association, they are no more soluble in the protein, they are no more soluble in the solvent mixture, so they could precipitate down. So this overall process is called immunoprecipitation. So we know what is precipitation. Precipitation is something where two different charges, when joined together, they make, for example, positive and negative charge, when joined together they make a salt, and salt is no more soluble in water. So you could uh, uh, affect the solubilization of the protein. So in other words, immunoprecipitation means you wanted to separate soluble protein, soluble fraction of the protein from a solvent mixture. So that is why you are now saying them immunoprecipitation technique. So in this case, you could say that this is the chromatin immunoprecipitation, because you are focusing on chromatin aspect of this overall thing. So you could couple this technique. So once you have done this chromatin immunoprecipitation, so you have an antibody against chromatin, any chromatin. So for example, it could be antibody against any of the four histone proteins, H3, H4, H2, H2B, and it could be an antibody against any modification on these histones. For example, modification on histone H3, modification on histone H2A, or any other. So this could be anything जिनके अगेंस्ट आप मालूम कर सकते हैं. So this is chromatin immunoprecipitation. So once you have done the CHIP technique, chromatin immunoprecipitation technique, so please pay special attention to to its denomination, because it's something different than this chip which is down there. I'm coming back to that. So you could do chip technique, and after doing this chip technique, you could couple it with another technique which is called maybe a simple PCR, or you could be more interested to quantify this PCR, so you could use qPCR. So this is chip qPCR. So if you know the target gene, you have to you can select that target gene, and you can amplify that target gene through this PCR mixture. And we know what PCR do. PCR is normally a technique in which you have two primers which could localize your gene of interest, and they will make amplification, and you will have your gene of interest by the end of the day. So this could only work if you know your target gene. If you do not know your target gene, so that, for example, you were interested, I mean, let me give you a scenario. You are interested to see if gene X, let's, any, this could be any gene, this, if you are interested to know whether your gene X is somehow uh, arranged in that particular chromatin fraction or not, if this particular gene is present in this chromatin fraction or not, or you could go for this target, you could go for this PCR. So now you need to have primers for this gene, which is X gene. So you have two primers for X gene, and you have already done this chip technique, and after doing this chip technique, you have fractions of this chip technique, and this will be the protein and DNA fraction. You will use this DNA for doing the PCR. I'm going coming back in next slide for the detail of chip technique. So there is a second technique which is called chip on chip technique. So if you do not know your target gene, this is a big question. So what will you do? You would like to see the genome wide chromatin maps in order to know what type of genes were present in this chromatin mixture which you have in your extracted chromatin. So for that purpose, you could go for chip on chip technique, or you could go for CHIP-seq technique. So these are two different techniques which will go for genome wide analysis. For example, the chip on chip means there is a chromatin immunoprecipitation coupled with this chip technique, which is a microarray. So you know what is, there is a small chip on which there are probes, and these probes are against multiple different genes or multiple different locus on your genome. So it could be 23,000 genes located on this chip. So you have this chip extract, and you overlay this chip extract on this chip, and by the end of the day, you will highlight some, you will get some fluorescence, and this fluorescence will be read by a computer, and which tells you, for example, if there were uh, that number of gene, let's suppose these are different probes जो कि उस चिप के ऊपर लगे हुए थे. मैं एक चिप अगर बना ही दूं आप लोगों के लिए, so it could be possible by the end of the day, after overlaying this extract, you could highlight this fraction. You will get maybe some fraction of on this area, you could have fluorescence on this area. So this will tell you that your chromatin extract contain these genes which are somehow could be identified through this technique. So by the end, you could identify genome wide loci which are present in your chromatin extract. Similarly, you could go for CHIP-seq. Now we have Illumina sequencing, third high throughput technique, in which you could use this overall chromatin extract and go for Illumina sequencing, and you will get genome wide reads, and which tells you that which loci were present in this gene. So this is a very important technique for studying chromatin research. So in chromatin research, a chip is a very important technique, which is in fact the starting, which gives you the starting material for whatever experiment you wanted to proceed. For example, you wanted जी
सर ये चिप का आपने बताया है प्रोब्स का, यानी दोनों टेक्निक्स में चिप टू चिप और चिप सीक्वेंस दोनों में प्रोब यूज होते हैं? अगर यूज होते हैं तो पहले से इसी तरह ये डिजाइन होते हैं कि होल जीनोम की क्रोमेटिन हम इस पे यानी डिटरमाइन कर सकते हैं?
ओके जो चिप ऑन चिप है उसके लिए तो फॉर एग्जांपल रेडीमेड चिप्स अवेलेबल एंड आई मीन इफ यू आर इंटरेस्टेड इन knowing what type of cancer related genes are maybe present in your chromatin extract, so there are already commercially available chips which is a pool of genes which maybe are involved in developmental processes. I mean, you could be interested to know, for example, you wanted to know the targets which are important for example, immune system. So there might are neurosystem. So there are chips which are commercially available. So there will be probe. But for CHIP-seq, normally we do not have specific probe. There are universal adapters which do not care about any sequence of genes. So that means they will create different reads. So there is no specific, for example, probe for this CHIP-seq. So if those who know what is the NGS, and they know that for NGS it is different than Sanger sequencing, in which for Sanger sequencing you know a specific primer sequence which could elongate just like PCR reaction. But in Illumina sequencing you do not know, you do not need that particular. You have only adapter sequences, and these sequences will amplify any gene or loci which is present there. Okay, I hope this could have answered the question. So there is no specific primers which are present for CHIP-seq. So there are limitations to these techniques. So for example, one of the limitation is that you always need a specific antibody to perform chip. I mean, if you do not have a specific antibody, you cannot make this experiment to happen. For example, there are commercial companies which could generate any antibody of your interest, so you could go for these type of chip analysis. Secondly, uh, inherent bias of localization could be possible, for example, because this technique required different steps, multiple steps, and at each step you could be making different types of biases. And more especially when you talk about this chip qPCR, it could be possible the gene of interest you are looking for, it is just a single copy gene. In that case, it is quite uh, difficult. It could be possible that your chip do not able to highlight that. You could not give you any positive result for that technique. Or on the other hand, if you go for CHIP-seq, you could get multiple reads, because if that gene of interest is duplicated at present twice or thrice, more than 2 times copies of that gene are present in your genome, it could give you different. So you could have false positive negative results based on this. So these are kind of limitations which are present everywhere. Secondly, you could go for mass spectrometry if you are interested in chromatin immunoprecipitation, in studying chromatin. So there are multiple mass spectrometric strategies available for that. Especially we might be interested in mass spectrometry of histones. For example, we know that there is a particular molecular weight of histone proteins, and we wanted to know if this histone protein somehow has been gone through any modification, so you could go for mass spectrometry. So what mass spectrometry will do, it will separate different entities of these histone based on their molecular mass to charge ratio. So for example, this could be an unmodified histone protein, it could be a modified histone peptide. So for example, and the difference of molecular weight between this histone and this histone might be equivalent to this CH3C double O moiety. So this will tell you that this histone protein is somehow modified. So I'm writing with through my so forgive forget if it's not beautiful. I'm not interested in beautiful writing currently. So you see, अगर तो इन दो हिस्टोन प्रोटीन्स के अंदर जो डिफरेंस है वो CH3C00H के करीब है, so इसका मतलब है कि this histone is somehow modified. So you could see that uh, that you could go for this technique as well to study different histone protein, different modifications. Recently, there is a recent developments about the in chromatin research is in chromatin conformation capture. So these are relatively new techniques which are called 3C techniques, or you could go for 4C technique, 5C techniques, high C technique. So 3C mean chromatin conformation capture. So you see there is a 3C. So that is why it is called chromatin conformation capture. So basic idea is to document chromatin in its spatial arrangement. This is a very important concept nowadays. You could imagine chromatin in linear order. So this is something like this, but this linear order will not give you much information about this chromatin, in what direction this chromatin. So it could be possible this point where my cursor is currently, for example, this point on chromatin on I have drawn a circle here. So for example, this chromatin which encompass two different strands, one is this one and other one is this one, it seems that this current cursor that they are present at one single location. So you see my fingers, focus on my finger, so this it seems like this. But if you look at it in three-dimensional space, it could be something like this. You understand my point now? So what does that mean? That means the three-dimensional space, three-dimensional property of chromatin is very important currently, and we know that because a cell is not a two-dimensional thing, cell is in fact a complete three-dimensional thing, and all the organelles of the cell are three-dimensional. So is the nuclei, and within the nuclei there is a chromatin, and this chromatin is somehow arranged in time and space environment in this spatial arrangement. And this technique is to capture that current conformation of the chromatin. So you see this focus, this word which is conformation, that means it is the three-dimensional structure of the chromatin. So there are techniques which are trying to uh, document, which are trying to enlist the three-dimensional structure of the chromatin. That is why these are called chromatin conformation capture technique. And because this is the simplest one of them, this is the simplest one of this technique, that is why it is called 3C technique. The most advanced form of these techniques are called 4C technique, which is 5C technique and high C technique. 3C technique in general term uh, could be used to study interaction between two locus. So for example, if this is one locus and this is a second locus, you could say this is an this is an X gene, this is an X locus, then this is a Y locus. Maybe if this words X and Y making problem to you, just think this is a locus and this is a B locus, and which is present, and both loci are present on your chromosome, on your genome, and you wanted to know at some particular time point whether these two loci has some interaction among each other or not. So this is a very important point which I'm going to tell you, and this is the our current understanding about the TAD boundaries. And you know what is TAD? These are topologically active domains. Topologically active domains, the term means within chromatin, not whole of the chromatin is simultaneously active, rather parts of the chromosome act as an active uh, entity, whether rest of them remained silent. So if you wanted to know two loci, so you could go for chromatin conformation capture. So what is the idea behind that? If in time and space, for example, these two locus are present on far apart in a chromosome, in a gene, maybe in genome. For example, this is the locus A and this is locus B, and these two loci, and in between them there are multiple different genes, millions and billions of bases, and it could be possible these two loci are present on two different chromosomes. I mean, do not fix yourself to this point that these are present on this particular chromosome only. So they could be present on two multiple chromosome, two different chromosomes. So these are two, and you wanted to know whether, and it happens to know that, I mean, these two are involved in some certain pathway. I mean, let's make a different thing. So you know that these two are involved in this in a certain pathway, but we wanted to know how possibly is it possible that the two are interacting in real time. So you could go for this technique. So you could capture the chromatin in real time. At that particular time point you will get the spatial arrangement of the chromatin, and by the end of the day, as you know that this is the gene A and this is the gene B, you could maybe get primer for gene A and a primer for gene B, and you wanted to amplify them. In a normal PCR, it is not possible. Let me tell you. In a normal PCR, this is not possible. Why not possible? Because if they are present on two different chromosomes, simply impossible. If they are even they are present on single chromosome, or if they are present on the single DNA strand, the distance among them is millions of bases. A normal PCR reaction may not amplify more than, for example, 10 kb size. So maybe not normally it is not possible, maybe for more than 10 kilobases with a normal PCR. So that means it is not possible in normal scenarios to amplify having one primer in one gene and the other primer in another gene, because they are not possible physically. They are not possible because they are so separate from each other, or they are present on two different genes on two different chromosomes. But if they are actually interacting with each other, if they are actually interacting with each other in time and space manner, just like this, as my fingers are now telling you at one point, and now they are in fact present spatially different point. So if they are interacting in time and space manner, you could capture that thing in through chromatin conformation, and you can fix that thing at that particular time of the particular moment of time. It's just like clicking your camera and taking a picture. So it might be at that particular time, uh, you are in a you are not in a good mood and you may be somehow very sad, but when the camera make a click, that particular time will be safe. You understand the point? So this is a similar thing for this gene. Normally it is not present in close vicinity, but because of chromatin conformation, chromatin somehow folding, they have come close to each other. So now when you will do PCR mixture, so you have fixed that thing, and after fixation you will extract your chromatin, just maybe again with this chromatin immunoprecipitation, and you can go for PCR reaction, and you will know that whether your gene of interest are interacting with each other or not. If they are interacting with each other or not, this will be a new information for you to understand. Being present at so far distance, still they could come close to each other. This is a very important information for you people now. So if you know the gene of interest, you could go for 3C. If you So this is called 1 versus 1. 3C mean 1 versus 1. So one locus versus one locus. So you know the information of both locus. 4C is an advanced technique, which is one locus, other locus. Sorry, just let me complete. 5C mean one locus with many locus. High C means one versus all. So these are multiple techniques. So if you know just one gene of interest and you do not know any other information, you could go for high C. you could go for 5C. But if you know one gene and one gene, you could go for 3C. So understand this. जी आप क्वेश्चन पूछना चाहती हैं जो
सर मैंने ये पूछना था कि हम हिस्टोन्स मॉडिफिकेशंस को स्टडी कर रहे हैं ना, तो हमारे पास कोई भी जो हिस्टोन की मॉडिफिकेशन होगी, मिथाइलेशन होगी या एसिटाइलेशन होगी, हम उनके लिए किसी फ्लोरेसेंट टेक्निक हमने अभी तक कोई यूज़ नहीं की या हम कर नहीं सकते, को हम फ्लोरेसेंट टेक्निक्स के थ्रू भी तो डिटेक्ट कर सकते हैं। मे बी कोई एसिटाइल ग्रुप हमें किसी टाइप की फ्लोरेसेंस अगर दे, तो आई जस्ट wanted to know is it possible or not?
Yes, exactly. All these immunoprecipitation techniques could be used for colorimetric analysis. For example, you could go for ELISA. I mean, this will create a color. This will create a fluorescence. So I mean, you could go for fluorescent microscopy techniques. I mean, if you, I'm going to show you maybe in next slides how you could do that. So if the only trick is you could couple your antibody and antigen, or you could couple your antibody of interest with relevant fluorescent dye, and it will create a color. So
करनी दोबारा रिपीट करें 3C हम उसके लिए यूज करते हैं सिंगल जीन के लिए बिटवीन टू लोकस सर 4C और
मैं आप लोगों को पूरी एक असाइनमेंट दे रहा हूँ इसके ऊपर, आप लोगों को मैंने क्योंकि पूरी डिटेल नहीं बताई आप लोगों को इसके ऊपर, मैंने जस्ट आपको बेसिक मिक्सचर बताया. सो वंस मोर 3C इज 1 वर्सेस 1, ठीक है. 4C 1 वर्सेस मैनी, 1 वर्सेस मैनी. 5C 1 वर्सेस all. High C all versus all. It's all type of interactions. It's all type of interactions which could be going on in chromatin. You could see the beautiful thing, the very important thing, it's the essence of our current understanding of biology, is that we are trying to think in a manner that the chromatin, which is in fact a linear entity, when fold together, could make a new networks of gene expressions. This is a very beautiful thing, which if you could imagine, what I wanted to tell you is that once the chromatin has been folded in three-dimensional space, it brought about so far located genes, so close to each other, they could work together in this closed environment. This is the essence of our current understanding